Hello everyone, I hope you all are doing well. So as I have mentioned, I am starting this series to discuss certain topics which are very much important, not only exam purpose. This will also help you in your interview for different integrated PhD programs or MSc programs in research institutes like IISc TIFR NCBS or different IISers. Now I will try to include certain things which are taught in the class here at IISc. So if you are lucky enough, you can get the exact same question in the interview as well. But this is absolutely not the purpose of this series. Now today I am going to discuss a topic which you might be asked if you are asked a question from microbiology. Now you are given a certain antibiotic which inhibits or kills bacteria, and very less information are available about that antibiotic. So how you will find the mechanism of action of that particular antibiotic? So this video is going to be about how to find out mechanism of action of an unknown antibiotic. Now to know the mechanism of action, you will be going to perform certain phenotypic, biochemical, genetic or molecular assays, and with that assays gradually we will get certain inferences, and finally we will conclude the mechanism of action. Now first of all, we need to know what is the mechanism of the antibiotic, if it kills the bacteria or it only inhibits the growth of the bacteria. So for that purpose we need to perform a time kill assay which will detect the time dependent killing of microbe. So we will plot log CFU per ml, that is the count of number of bacterial colonies versus time. So after the application of the drug, how the number of colony changes. So if the drug is a static drug, if it only inhibits the growth of the bacteria, we will get a straight line, there will be no growth, we will get a straight line. But if it is a bactericidal drug, if it kills the bacteria, the number of colonies will decrease. Therefore, if with the time kill assay, if we get a straight line, this is a bacteriostatic drug, and if we get a curve which is slightly declining, like sharply declining, then this will infer that the drug is a bactericidal drug. Now after determining this, we need to know the MIC and MBC value, that is the minimum inhibitory concentration and minimum bactericidal concentration. So what are those values? Now MIC value is the minimum concentration or lowest concentration of an antimicrobial agent or antibiotic which prevents the visible growth of the microorganism after a certain incubation period, generally overnight. So we are adding the antibiotic in the culture media and growing the bacteria and incubating the culture for a certain period of time, generally overnight. And the lowest concentration of the antimicrobial agent which inhibits the visible growth of the microorganism is called the minimum inhibitory concentration. Now what is the definition of minimum bactericidal concentration, or MBC? MBC is the minimum concentration or lowest concentration of an antimicrobial agent that kills 99.9% of the initial bacterial inoculum or initial bacterial population after a specific incubation period. So MIC basically deals with the inhibition of visible growth, like if we are growing bacterial culture in a liquid culture media, in a broth, then if there is bacterial growth, there will be turbidity in the medium. We will get certain absorbance value in OD 600. Generally we measure the bacterial growth at 600 nanometer. So we will measure the absorbance at 600 nanometer, and if we get a certain value, there is visible bacterial growth. But after adding a certain concentration of antibiotic, if there is no growth, then that minimum concentration will be the MIC value, and for determining MBC value, the visible growth is not sufficient. We need to confirm that 99.9% bacteria are killed in the media. Now you might think that if there is no visible growth, then bacteria are killed. That's why there is no visible growth. But that's not always the case. Now let me discuss something. How to determine MIC and MBC values. Now to determine MIC values, as I have mentioned, we will add different concentration of antibiotic, like here is 1 microgram per ml, 2 microgram per ml, and we gradually increase the concentration. 1, 2, 4, 8, 16, 32, 64, 128, 256, 512. These are the concentration of the antibiotic is gradually increasing, and you can see here at the concentration of 16 mg per ml there is no visible growth. Therefore the MIC value is 16 mg per ml. So till 8 mg per ml there was growth, but when the concentration becomes 16 mg per ml there is no visible growth. Therefore the MIC value is 16 mg per ml. But to detect the MBC value we need to culture bacteria from these tubes in a drug-free agar plate or drug-free culture media. So these are the drug-free media, drug-free agar plate. There is no drug, there is no antibiotic in these plates, but in these tubes there is antibiotic. Okay, now here we have seen that till 8 microgram per ml there was visible growth, but whenever the concentration becomes 16 microgram per ml or higher there was no visible growth. So if MIC and MBC values are equal, we should have visible growth, I mean we should have culture growth till 8 microgram per ml, but whenever the concentration becomes more than 8 microgram per ml, that is 16 microgram per ml or more, we should not have any kind of growth, right? But here you can see the case is slightly different. Whenever the concentration is 16 microgram per ml there was no visible growth. Here you can see there is no visible growth, but there was growth in drug-free agar plate. Therefore 99.9% bacteria are not killed. Might be 95% or 96% bacteria are killed. Therefore there was like no visible growth, but that 2 or 3% bacteria which are still alive in 16 microgram per ml are able to give colonies in drug-free agar plate. Therefore to detect the minimum bactericidal concentration or MBC value we need to again culture the microbes in a drug-free agar plate. So to determine MBC, would you detect the visible growth? No. To determine MIC we detect the visible growth, and to detect MBC value we need to culture the microbe in a drug-free agar plate. Now this I have already discussed. Now we have several information about the antibiotic. First of all, we know if it is a bacteriostatic or bactericidal, and we also know the MIC and MBC values. Now we will perform several biochemical and molecular biology experiment to detect the target of the antibiotic. Now first of all, we can perform certain morphological analysis like microscopy techniques to detect the change in morphology of the bacteria. Suppose the antibiotic is targeting cell membrane or cell wall, then we can see swelling or lysis or leakage. So this will infer that the drug might be targeting cell membrane or cell wall. But if there is no such kind of leakage, but there was elongation of filaments or filamentous forms, appearance of filamentous forms, then might be the drug is targeting protein synthesis pathway or DNA replication pathway. Because if the protein synthesis pathway or DNA replication pathways are inhibited, the cells will not die immediately because the membranes are still intact, but the cell division will be arrested because there are certain important proteins like FTSZ which are important in cell division. So that proteins will not be synthesized and the bacteria will be unable to divide. But still, with the help of other proteins, the bacteria can grow. Therefore, there will be elongation of filaments and inclusion bodies. There will be formation of certain filamentous forms because of the growth. But because of the lack of certain proteins, the cell will not be able to divide. Therefore, If there is swelling or lysis, the drug probably affecting the cell wall synthesis pathway. If there is leakage or appearance of ghost cell, like a cell with everything leaked out, there is nothing inside the cell, then the drug is probably targeting causing membrane disruption. If there is appearance of elongated filaments or inclusion bodies, then the drug probably targeting protein synthesis pathway. But if there is the synthesis of filamentous forms, then probably the drug is targeting DNA replication pathway. And we can also stain the cell with certain dyes like propidium iodide, which can only enter the cell if the membrane is compromised, the membrane is damaged. So let's assume we are staining the cell with propidium iodide, and we are seeing that the cell is properly stained. That means the membrane is compromised. So the antibiotic is affecting the membrane synthesis pathway. So by this way, these are the preliminary techniques by which we can guess which might be the possible mechanism of action of the antibiotic. So in this diagram here you can see, here I have tried to explain, like in case of the first bacteria, the Salmonella enterica, with the application of polymyxin B, here you can see there is a change in the membrane structure or cell wall. So with the high concentration, like after 120 minutes, you can see there was a substantial change in the structure of the cell membrane or cell wall. So probably this drug is affecting the cell with disrupting the membrane synthesis of cell wall synthesis pathway. But in case of the other bacteria, you can see after 120 minutes there is no visible change in the membrane structure or cell wall structure. So the second bacteria is probably resistant to this particular antibiotic. So with the microscopic result or staining, we can get a preliminary idea how the antibiotic is performing. Now after that we will perform the macromolecular synthesis assay, which will confirm what might be the mechanism of action. So what we actually do, we will incubate the cell with radio-labeled precursor of DNA replication pathway or RNA synthesis pathway or protein synthesis pathway or cell wall synthesis pathway or lipid synthesis pathway, and after a certain period, around 30 minutes, we will stop the reaction and we will check how much radio-labeled precursor is incorporated. So let's assume that a particular drug is targeting the RNA synthesis pathway. Okay, now if the RNA synthesis pathway is compromised, there will be very less incorporation of radioactive uridine, and if the DNA synthesis pathway is compromised, there will be very less incorporation of radio-labeled thymidine. If the protein synthesis pathway is compromised, there will be very less incorporation of radio-labeled leucine. I hope this point is clear. So the particular pathway that the drug is targeting, that particular precursor will be incorporated in lesser amount. So I will show you a data such that you can understand well. So here you can see we are incubating the cell with 3 radio-labeled precursor: radio-labeled thymidine, radio-labeled uridine and radio-labeled leucine, that is an amino acid. And this is the control group, and this group is treated with drug, that is the antibiotic. And we are checking percentage incorporation with respect to the control group. Now in case of radio-labeled thymidine, you can see 98% incorporation. So if 10,000 if the incorporation is 10,000 ppm, ppm is counts per minute, which is unit of radioactivity. 10,000 counts per minute in case of the control group. In case of the treated group, the incorporation is 9,800, that is 98%. But in case of the leucine, the incorporation is 97% of the control group. But in case of radioactive uridine, the incorporation is only 13%. Therefore, the uridine incorporation pathway is hampered in case of the drug treated group. So we can say that RNA synthesis pathway is inhibited. So the possible mechanism is inhibition of RNA synthesis, as rifamycin does. Rifamycin particularly inhibits RNA polymerase enzyme, therefore transcription is inhibited. So in this way we can know we can get to know the exact mechanism of action of that particular drug. Now here I have added this bar diagram to explain better way. Now the gray color bar diagrams are with the unknown antibiotic for which we want to know the mechanism of action, and the white diagrams are antibiotic for which the action is known. Now for DNA, this is an antibiotic which inhibits DNA synthesis. For RNA, this is an antibiotic which inhibits RNA synthesis, like rifamycin. This is peptidoglycan. So for peptidoglycan, this is an antibiotic which inhibits peptidoglycan biosynthesis. For protein synthesis, this is erythromycin antibiotic which inhibits protein synthesis. And this gray color bar diagrams are basically antibiotic for which we don't know the exact mechanism of action. So for DNA, you can see for ciprofloxacin, which inhibits DNA replication, the amount of DNA is substantially decreased, but amount of incorporated DNA is substantially decreased. But for the unknown drug, the DNA incorporation has not reduced. For RNA, the amount of RNA incorporation is decreased in case of rifamycin because it inhibits RNA synthesis. But in case of the like unknown drug, the RNA incorporation has not decreased. In case of the protein synthesis, in case of erythromycin, it inhibits protein synthesis, therefore incorporation of radio-labeled amino acid has been decreased. But in case of the unknown drug, there is no substantial decrease, like almost near to 100%. But in case of peptidoglycan, as in case of vancomycin, which inhibits peptidoglycan biosynthesis or cell wall biosynthesis, the incorporation is decreased, and for the unknown drug also, the incorporation of peptidoglycan is also decreased. Therefore, the unknown drug and vancomycin shows similar mechanism of action. Therefore, both of them exhibits inhibition of cell wall biosynthesis. So in this way, we can get an idea how an antibiotic actually functions, what is the possible mechanism of action of an antibiotic. Now we don't need to do all the experiments if we have a resistant strain for that particular antibiotic. Suppose we found a particular antibiotic and we have also found a certain bacterial strains which are which are resistant against that particular antibiotic. So we can compare the genome of the resistant strain with a susceptible strain, and we will find certain SNPs, certain mutations in the strain of the resistant bacteria, and suppose we are getting 10 SNPs. So we can screen them, and possibly a certain protein within that 10 proteins will be the possible target of the drug, and that proteins are mutated in case of the resistant strain. That's why the drug cannot inhibit the growth of the bacteria in case of the resistant strain. So we need to give less effort if we find a bacteria or microorganism with a resistant strain, because we can compare the genome of the resistant strain with a susceptible strain, and we can narrow down the possible targets, and we can confirm whether this protein or that protein or that component is basically the target of the particular drug. So how we can do that? We can perform overexpression or knockdown experiment. Suppose there were 10 mutations in the resistant strain. There were, suppose, protein A, protein B, protein C, protein D, and in that way. First of all, we are overexpressing protein A in case of the susceptible strain, like wild type strain. And if protein A is the target of the antibiotic, and after overexpressing protein A, the susceptible strain will be resistant will be resistant to that particular antibiotic, because there will be more amount of protein A, and to inhibit their growth we need to supply more antibiotic. Otherwise that same amount of antibiotic will be unable to inhibit the growth of the microorganism. So overexpression, so if by overexpression that particular protein we get a resistant phenotype, then we can infer that that is the target of that particular drug. And by knockdown, by reducing the amount of protein A, if the bacteria becomes more sensitive to the drug, then also we can tell that protein A is the target of that particular antibiotic. So initially this was the amount of protein A, and by overexpression we are increasing the amount of protein A, and by knockdown we are decreasing the amount of protein A. So in the first case we will need more amount of antibiotic to inhibit the higher amount of protein A produced by overexpressing. Therefore the bacteria will be resistant to protein A, because we have to provide the bacteria with more amount of protein A. And in the second case there will be the lesser amount of protein A, therefore a smaller amount of antibiotic will be able to inhibit the bacteria, because the amount of protein A is already reduced, and a smaller amount of the antibiotic will be able to inhibit the bacteria. So by overexpression or knockdown experiments we can validate if the hypothesis about the 10 proteins we are initially giving, which one is correct. Like if overexpression makes the bacteria resistant and knockdown makes the bacteria more sensitive, then we can confirm that this is the target of that particular antibiotic. So to summarize everything, first of all we will detect the MIC value, and with time kill assay we will detect the nature of the antibiotic, if it is a bactericidal or bacteriostatic drug. Then we will perform microscopy techniques to initially assume what might be the mechanism of action through observing microscopic changes in the structure. Then the most important part is the macromolecular synthesis assay, through which we will detect the incorporation of certain precursors. So if the precursor of DNA synthesis is not incorporated in higher amount, or the incorporation is decreased, then the drug is possibly targeting the DNA synthesis pathway. And if we have any resistant strain, we will perform resistant mutation screening, and we will infer which might be the possible target proteins, and we will validate our result by overexpression or knockdown experiments. Now I hope the techniques which I have mentioned seems logical to you. If you have any further question, you can definitely ask me in the comment section, and I will like to get your feedback. How was this video, such that I can improve, or I can make separate different kinds of videos on different topics of this same series. So thank you very much for your patience, and if you have any questions, certainly you can ask me in the comment section. I will definitely try to reply as soon as possible, and thank you very much.